Hello, everyone. Thank you so much for joining us today. My name is Afrida Rahman Enyart. I am a scientific liaison here at Protein Tech. And today I'm going to be talking about nanobodies and how they're ideal for IP and IF workflows. So during the presentation, if at any point you have any questions, please type them into the chat box or into our Q&A box. So I want to begin by talking a little bit about the company. So Protein Tech is a leading manufacturer of antibodies, immunoassays, and recombinant proteins. We also have antibodies against more than 13,000 targets. We also offer ELISA kits, IHC kits, and cell separation kits. In 2020, Chromotech became a part of the Protein Tech family. And Chromotech develops nanobody-based reagents for immunoprecipitation, immunofluorescence, and for protein purification. And the third product line that we have as a part of Protein Tech is Humankind. And Humankind manufactures cytokines and growth factors, and all our proteins are expressed in human HEC293 cells, meaning that they have native folding, post-translational modifications, and glycosylation. And we also offer both research use and GMP grade products. So what makes Protein Tech different from other companies is that our products are manufactured in-house and sold only by us with our Protein Tech label. So you won't see our products under any other company name. We also offer full transparency about our products, so we share detailed reports about our product performance, including original validation data and quality control. So today I want to talk about our Chromotech nanobody products. So we have a diverse product portfolio to aid in immunoprecipitations, immunofluorescence, and protein purification. And today I want to highlight three product types that can really enhance your IP and IF workflows. So we will cover nanotraps, which are ideal for immunoprecipitations, nanosecondary reagents, which are ideal for immunofluorescence, and our spot system, which is a one-stop shop that can aid in various workflows. So before I dive into our nanobody-based products, I want to talk a little bit more about what nanobodies or VHHs are. So as you probably already know, a traditional antibody is composed of a heavy and a light chain. But alpacas and other cameloids, in addition to having this traditional type of antibody, also have another type of antibody which consists of a heavy chain, and this is called a heavy chain antibody. So the binding domain of this heavy chain antibody shown here in green is what we call a nanobody. And the nanobody is very small, it's only 15 kilodaltons in size. And then we can take this nanobody and we can conjugate that to a bead, which we call a nanotrap, or we can conjugate it to a dye, which we call our nanosecondary reagents. So nanotraps are ideal when you're trying to conduct immunoprecipitations, whereas our nanosecondary reagents are optimal for immunofluorescence. So I'll start by talking more in depth about our nanotraps, which are our nanobodies that are conjugated to a bead. So we have nanotraps against GFP, which will be my focus for today, and that's what we call our GFP trap. But we also offer nanotraps against other targets such as M-cherry, RFP, or peptide tags such as V5. So our nanotraps are ideal for immunoprecipitations, and so I'm sure most of you are familiar with IP, but for those who aren't or who haven't done it in a while, IP is a technique where you can isolate a protein of interest out of a lysate using an antibody that binds specifically to your protein of interest. So generally you have your antibody that is coupled to a bead, and then it can bind to your protein of interest, and then you can wash away any unbound proteins. And this then allows for an enriched sample of your protein of interest and any of its interaction partners. So you can then process these samples and utilize them for something like Western blotting. So here we're looking at a comparison of our GFP trap compared to doing a conventional antibody pull down. So in a traditional IP, there's a pre-incubation step in order to immobilize and bind an antibody onto protein A or G beads. The nanotrap, on the other hand, is ready to use with the bead, and it's already conjugated to the nanobody. So there's no pre-incubation step. All that is really required is a quick wash step, and then you can start your immunoprecipitation. So now we'll look over here at the SDS page to see how the GFP trap and the conventional antibody perform after the IP. So the three lanes here on each of the gels shows the input of your cell lysate, the flow through, which is essentially the supernatant after you've conducted your IP, and then the bound fraction, which should consist of your enriched protein of interest. So we can see that the bound fraction here in the middle when using both the GFP trap and the conventional antibody. But you can see that after elution with the SDS sample buffer, we not only see our protein of interest, in this case GFP, but we also see our heavy and light chains that are released from the beads and show up on this SDS page. And what's really more striking is that the bands for the heavy and the light chains show up even brighter than the protein that you're interested in looking at. So sometimes you might end up playing this guessing game of whether a band is actually your protein of interest or if it's a part of the antibody. But now if you look at the left with the GFP trap, because the nanobody is so small, it doesn't show up on SDS page. Also, because the beads are covalently bound to the nanobody remains bound to the beads even after elution with SDS sample buffer. And therefore, you only get one single band in the SDS page, which is going to be your protein of interest. The other great aspect of using a nanobody is that nanobodies bind their targets very tightly. They have a very high affinity, which leads to a very effective pull down, even when your protein of interest is expressed at low amounts. So for example, when you look at the Western blot over here, you can see that there's essentially no protein of interest that's left in the flow through when using the GFP trap, and all of the protein is enriched here within the beads. When you compare that with a traditional IP, you can see that you do see an enrichment of protein in the bound fraction, but there's still some that's left over in the flow through. So another benefit of the GFP trap is that it is faster and more efficient than traditional IP. So as I mentioned previously, in a conventional IP, you have your cell lysis, which you need to incubate with your target antibody, and then couple with protein A and G beads. You then need to wash the bound beads prior to elution with SDS sample buffer. And this process in the end can take more than 3 hours. However, with our GFP trap, since the nanobody is already coupled to the bead, you can actually directly incubate your cell lysis with the GFP trap, wash, and then go ahead and elute. And this process can take less than 2 hours. So on the right hand, I have a blot showing just how quickly our GFP trap works. So within 1 minute of incubation, we already see our protein of interest here in the bound fraction. And by 30 minutes, we see complete binding of our protein of interest with non-detectable amounts of protein in the flow through. So we do recommend that when using GFP trap that you incubate for an hour and use about 25 microliters of the GFP trap. So as I had mentioned earlier, our GFP trap has very high affinity. In addition to this, the binding potential is also very stable. So here what we're looking at is our SDS page with samples using different wash buffer detergent conditions. And as you can see, even when using 2% NP40 or 0.2% SDS, this doesn't affect the binding of GFP trap to your protein of interest. GFP trap binding is stable in many different buffer conditions, including buffers with reducing or chaotropic reagents, high salt concentrations, or even harsh detergents or glycerol. So we want to make sure that our GFP trap fits the needs of our customers. So we have this nanotrap conjugated to different types of beads, depending on what you need for your IP. So this includes agarose, magnetic agarose, and M270 magnetic particles. And the type of bead that you choose can depend on a few things. So for example, we recommend using the M270 particles if your protein or complex is larger than 200 kilodaltons. However, if your protein or complex is less than 200 kilodaltons, we recommend going with agarose beads because they tend to have a higher binding capacity and generate lower background. But whether you go with the traditional agarose or magnetic agarose is a personal preference. So if you're interested in conducting an IP and would like to try a nanobody, we do offer nanotraps against several fluorescent tagged proteins such as GFP, M neon green, M cherry, etc. We also offer nanotraps against other small peptide tags, a couple solubilization tags, and a few oncology proteins. But if you're looking to conduct an IP for other endogenous proteins, our protein tech catalog does have over 4,000 antibodies that are validated for endogenous targets, and over 3,800 of these antibodies are validated for IP. We also offer antibodies against 13,000 targets for Western blotting. So now that I've talked a little bit more about our nanotraps, specifically our GFP trap, I want to move on to talking more about how you can optimize an immunofluorescence workflow. And you can do this by utilizing nanosecondary reagents. So our nanosecondary reagents are also nanobodies, but what makes them different is that they are conjugated to fluorescent dyes. And our nanosecondary reagents target both, can target mouse or rabbit IgG antibodies. So the main benefit of using nanosecondaries is the faster staining protocol. So we all know that time is money and that your time is very valuable. So with a standard protocol, you need to first fix, permeabilize, and then block your cells. And then you do your primary incubation for about an hour. You do your washes, then you do your secondary antibody incubation. You wash that off, and then you can image. So this protocol takes about 140 minutes. But with the nanosecondary reagents, you can do a one-step staining. So because the nanosecondaries are monovalent, you can actually incubate your primary antibody and the nanosecondaries with the sample at the same time. So with the nanosecondaries, you can fix, permeabilize, and block your cells, and then co-incubate your primary antibody and nanosecondaries for an hour, do your washes, and then image. And so this then cuts the time it takes to do your staining in half. So here on the right, you can see a comparison between one-step staining with our nanosecondaries versus using a traditional staining protocol with a standard workflow. And you can see that with one-step staining, this works just as well as if you did sequential staining. So there's really no difference and you get the same quality image, but you can save half the time. Also, because there is only a one-step incubation, the staining is more hands-off. And so incubation of these samples ends up being more gentle. And gentle incubation is ideal when you're trying to conduct something like flow cytometry, where you are doing live cell imaging. And it also is great for multiplexing. The other great thing about the nanosecondary reagents is that it allows for a higher resolution because the nanobodies are so small. So the nanosecondaries are about 10 times smaller than a conventional IgG antibody, and this leads to a different staining pattern. This can be seen in this cartoon right here that shows the small epitope label displacement of our nanosecondaries. So we have our target shown here in purple, and then the primary antibody that recognizes it shown here in gray. And then you have the nanosecondaries shown here in green that bind very closely to the antigen because of its small size. In comparison to a polyclonal secondary antibody, you can see that the dyes in the end are much farther away from the antigen and the target, which leads to a lower resolution. The smaller size also allows for better tissue penetration. So here we have two examples. One is a STED image where you can see it's very high resolution. And here on the right, we have a customer image. It's a 3D sim image. And again, you see that's very high resolution. This customer also measured the resolution of our nanosecondaries compared to traditional IgG secondaries and concluded that our nanosecondaries, you could gain about 20 nanometers in resolution. And this higher resolution is important for IF, but definitely for other super resolution microscopy such as STORM. The third benefit of our nanosecondary reagents is the homogeneous sample staining. So because of the small size, the nanosecondaries can diffuse faster and more homogeneously into tissues or dense cell compartments. So this can be seen here in these images from a customer. They compared our nanosecondary with a conventional secondary antibody. And you can see that the conventional antibody, you have some bright staining here in the outer layer, and then some spots over here. But the staining with the nanosecondaries is much more homogeneous, especially here within the inner layers. So this homogeneous sample staining is very important for IF because you want to make sure that you're accurately imaging your protein of interest within your tissue and reporting the correct localization of these proteins. Another benefit of our nanosecondary reagents is the low background, no cross-reactivity, and high subclass specificity. So all our nanosecondaries are tested for their subclass specificity, and this can be seen right here in this blot. So our mouse IgG1 nanosecondary binds only to mouse IgG1 and not to other IgG subclasses. And this is the same for all our other nanosecondaries. We compared this with a competitor secondary antibody, and this secondary antibody was supposed to recognize rabbit IgG, but this secondary antibody still recognized other mouse IgG primary antibodies. So here we have our nanosecondaries with no cross-reactivity and low background. And this is very important for something like multiplexing. So here on the left, we're showing an image where three mouse primary antibodies of different subclasses were used with three different nanosecondaries. And you can see that this is a great quality image with very low background and very specific staining with no cross-reactivity. Our nanosecondaries are very well characterized, they're reliable, and they're very thoroughly validated. And this is important because you don't want to be spending your time and money with subpar antibodies. So we do validate our nanosecondaries for specific applications. We test the subclass specificity for each nanobody clone. We test the cross-reactivity. Our nanosecondaries are recombinantly expressed, so we essentially have no lot-to-lot variations, and we have an unlimited supply. We have also determined the affinity, the melting point, and the degree of labeling as well. So just an overview of our nanosecondary reagents so far. Currently, we have nanosecondaries that are specific for rabbit, which are also co-reactive to human IgG. So some of our customers actually use our rabbit nanosecondaries against human antibodies. We also cover 3 of the 5 subclasses of mouse IgGs, including IgG1, IgG2b, and IgG3. Our nanosecondaries are monoclonals that are either Fab or FC specific, or in the case of our rabbit nanosecondary, which recognizes both. Our nanosecondaries are also conjugated to Alexa dyes, specifically 488, 568, and 647, but we can also offer the unconjugated clones on demand. And our nanosecondaries have been utilized by customers successfully who do IF, flow, and Western blotting at relatively good dilutions. So finally today, I want to discuss our spot system, which uses a spot tag. And a spot tag are these 12 amino acids right here. And we provide nanobody products with this tag conjugated in different ways based on what technique you are utilizing. So our spot system products are similar to our other product categories, except for the fact that they recognize a spot tag. So similarly to our nanotraps and nanosecondaries, we take the nanobody and then we can conjugate the nanobody in a bivalent format and conjugated to a fluorescent dye for IF, as well as conjugated to beads. So we have our spot trap, which is ideal for immunoprecipitations. And then we also have spot cap, which is ideal for protein purification. So as I mentioned, the spot tag is these 12 amino acid, as a 12 amino acid peptide tag that was developed and has shown to be superior to other more popular tags such as his tag or flag tag. The spot tag is it's very small and it's very easy to clone. So it can be tagged onto the N or the C terminus of your protein of interest. Also, this tag has been tested and it can be used in bacteria, it can be used in yeast, mammalian cell lines, plant cells, and insect cells. Our spot system products that incorporate nanobodies are also very stable in harsh conditions and buffers. They have a high capacity in many regeneration cycles, so this makes it cheaper than the flag tag. Our products are also applicable to various techniques such as IP protein purification, IF, and Western blotting. And similarly to our other nanobody products, we also see high affinity, better labeling and better label density and epitope access, as well as the ability for higher resolution imaging. So I'll start by talking about the spot tag and nanobody. So here in the green, we can see the nanobody, and the structure right here is the spot peptide. And you can see that the spot peptide actually goes through the nanobody. And this is done by clamping by glutamate and arginine residues that actually hold the peptide tightly to the surface of the nanobody. So the spot tag is derived from beta-catenin. It's embedded on the surface, and then it becomes a part of the beta sheet structure. And then, as I mentioned, the tag becomes locked by glutamate and arginine side chains. The spot nanobody can recognize the spot tag on your protein of interest, whether it's on the N or the C terminus. And the spot nanobody has very high affinity, high stability to buffers, and it also has high specificity to target the target peptide. Our spot system really is a one-stop shop. So you can use spot for various techniques such as super resolution microscopy, protein purification, immunofluorescence, Western blotting, immunoprecipitation, mass spec, and even CRISPR-Cas experiments. So I want to start talking about the spot system by talking about the spot trap. So the spot trap is like the other nanotraps in that it is a nanobody that is covalently coupled to a bead. And similarly, we don't have any heavy or light chain contamination that appears on either the SDS page or the Western blot. And the spot tag has a very high affinity, so it binds very tightly to the spot tag. You can also use the spot tag for IP from cell lysates of different species, and it even works when your protein of interest is expressed at very low levels. The spot trap also has very low background compared to other commonly used reagents for other peptide tags. So here what we did was we tested IPs using common reagents from different suppliers using HEC293T cell lysates that did not express any sort of tag. So after elution of each resin with SDS sample buffer, we ran the samples on SDS page. And if you look here on the first lane where we tested the glutathione cellulose from one supplier, you can see that there's a very high background, even though there's no GST expressed within that cell lysate. With the MIC and the flag reagents, you can see not only the high background, but you also see the heavy and the light chains. And then with the flag dynabeads, again, you see some background there too. So these last two lanes are our spot traps that are either conjugated to magnetic agarose or agarose. And you can see that there's very clean background with no unspecific staining. Spot trap also has a superior performance when it comes to the washing conditions. So we know that sometimes harsh washing conditions are required in order to remove all the unwanted proteins that might be unspecifically bound. So you can see that our spot trap works well even with wash buffer using high salt concentrations and high amounts of various detergents and even reducing agents or chaotropic salts. It also has a very high pH stability as well. So from pH 4 to pH 10, you won't see changes in any binding efficiency. So then I want to move on to the spot cap. So what's the difference between the spot trap and the spot cap? So the spot cap is very similar in that it is a nanobody that's conjugated to a bead, but this is used for protein purification purposes. So whereas our spot traps can only be eluted with SDS sample buffer, when using our spot cap, the protein can be eluted using either a spot peptide or with 100 millimolar of glycine. So here on the right, we are looking at SDS page, and we show here the input, the flow through, and you can see here that not all the protein has been bound, but after the wash, the protein can then be eluted with 100 micromolar of spot peptide, and we get a substantial amount of protein eluted, especially within elution 2, 3, and 4. And other than your protein of interest and some bits of degradation that is expected, you can see that this is a very clean gel for protein purification. The binding is also quite good. So we get about 10 mgs of protein per mil of settled resin. And it's also highly specific. So as I mentioned, you don't see any contamination on the gel. Our spot cap also has very low background when utilizing samples from multiple species. So here we incubated cell lysates without spot label with our spot cap beads and then washed away all our unbound protein and then eluted with spot peptide. And you can see that in mammalian cells, yeast cells, insect cells, and bacteria, we have very faint to no unspecific bands on our gel. And then here on the right, you can see a comparison between our spot cap beads with wild type. You can see a comparison of our spot cap beads with a competitor flag tag resin. So here we incubated our beads with wild type HEC cells expressing no spot tag. And then we washed away any unbound protein and then eluted with the spot peptide. And you can see essentially no background. But if you look at this gel over here, you can see when we similarly tested hex cells with no flag tag, incubated with flag tag resin, and eluted with flag peptide, there are all these host cell bands that bind to the flag agarose. We also have our spot label, which is another way to detect the spot tag attached to your protein of interest. And the spot label is ideal for immunofluorescence. So as I mentioned earlier, our spot label is a bivalent molecule with a fluorescent dye. And it recognizes your spot tag in your tissue of interest and it labels it with a fluorescent marker. So here on the left is a very nice example of confocal microscopy looking at TOM70 EGFP spot tag. And you can see that the EGFP signal perfectly colocalizes with our staining obtained by using our spot label. Additionally, here on the right, we have HeLa cells that express spot-tagged actin or our control staining without spot tag. And you can see that when using our spot label, we see high resolution staining that recognizes the spot tag, but essentially generates no background. So we have a product that is great for high resolution images and low background. We have also developed a mouse IgG1 antibody against spot tag that can be used for Western blotting. So we have observed that this antibody can detect spot tag that is expressed at either the N terminus shown here on the left or at the C terminus shown here on the right of your protein of interest. So the antibody can also detect the spot tagged protein at fairly low levels of expression as well. So then finally, I just want to do a brief overview of the spot system reagents that I talked about today because there were so many. So we offer the spot trap, which is our spot nanobody covalently conjugated to agarose, or magnetic particles. And this is ideal for immunoprecipitations. We have our spot cap, which consists of our spot nanobody conjugated to a bead that can be eluted with our spot peptide or glycine. And this is ideal for protein purification. We also have our spot label, that is our bivalent spot nanobody conjugated to fluorophores, and this is ideal for immunofluorescence. And we also offer the spot nanobody that is unconjugated if that is preferred. And of course, we have our spot tag antibody that is ideal for Western blotting. So to end this talk, I would like to share the success of not only us as a company, but our customers as well. So our customers have cited protein tech products in over 150,000 publications worldwide. And a recent paper in EMBO Reports by Magastrati et al. About myosin 6 used the Chromotech GFP trap for immunoprecipitation. We are also very proud that Protein Tech won the Researcher's Choice Award from SiteAb this year, which demonstrates that our customers really do value the quality of our products. So as an attendee of today's seminar, Protein Tech would like to offer a free sample of a Chromotech nanotrap or a free sample of any Protein Tech antibody. We'll provide this link to you guys in a follow-up email, so don't worry about having to copy it down or anything. So with that, I would like to thank everyone for attending today's webinar about nanobodies. If there are any questions that I cannot answer now in the Q&A session, please do feel free to email us at support@chromotech.com, or you can email us at events@ptglab.com, or you can email me directly at afrida@ptglab.com. Also, if you liked today's events and you want to know more about our webinars and our workshops, please do follow our socials because we post about them there. So with that, I'll go ahead and I'll begin the Q&A session. Okay. So we have a question from Fran asking, can you add DAPI with the primary and secondary mix? Yes, you can. That should not interfere at all. Is the nanosecondary antibody good for Western blot? So we've had customers who have used it for Western blotting before. We have not tested that in-house. So that's something that you could try a sample and see if it works in your hands. But we have had customers who have used it before. We also have, can the GFP trap be used for Western blotting? So we don't recommend it. The GFP trap is mainly ideal for immunoprecipitation. So we would recommend using the GFP trap for immunoprecipitation and then utilizing, a polyclonal antibody for Western blotting. What lysate volume or protein concentration is needed for GFP trap to detect your protein? So this is what makes GFP trap so great. There's no set, lysate volume. So if you have a protein that is expressed at very low amounts, you can actually use a lot of cell lysate. So you can use, 10 mils of cell lysate if you need to, and you only still need 25 microliters of the GFP trap because it can detect GFP proteins so well. So for generating a spot tag protein of interest, do you have a do you have plasmid templates or provide the sequence? Yeah, we can definitely provide the sequence. And if you do have any questions about cloning, you can definitely reach out to our Chromatech support team, and they should be able to answer any of your questions and kind of help you with that too. So I noticed that the duration of the primary and secondary nanobody was 1 hour. Is this typical? What happens when longer duration is used, say overnight? Is background an issue with longer time periods? Um, no, so it's just ideal to do it in 1 hour. I don't think we've had any background issues if you do it for longer. Um, but the whole idea is that you can do these protocols very quickly. But again, if you do your staining overnight, we just recommend doing it at 4 degrees. Can we use the GFP trap for pulling down ribosomes for RNA-seq? That's a very good question. I'm not totally sure if if any of our customers have done that. So that might be something that requires testing it. Um, I know we haven't tested that in-house. So again, if you wanted to get a sample and kind of try and see if it works, we also have a protein tech guarantee. So if you purchase a product and it doesn't work in the technique you want to do, and our technical specialists can't help you, then we will refund you. So it's kind of a low risk for you to try things out. Okay. Um, for the nanobody, does it contain an FC domain? Um, This I will get back to you on. I need to ask one of our Chromatech technical specialists, kind of the makeup of the nanobody. So I will definitely get you a response by email. All right. Are there any other questions? All right, well, with that, I want to thank everyone once again for joining this webinar. I hope you found it useful, and please do look out for that follow-up email. We will be providing the link for your free sample, and we will also be sending out certificates of attendance next week. So please do be on the lookout for that. All right, well, thank you, everyone. Enjoy the rest of your day.